Again on an oral application for a nanomilling case study by Dr. Olaf Behrend from Boehringer Ingelheim Pharma. Olaf Behrend is the Boehringer Ingelheim site as lab head of pharmaceutical development.
Okay, thank you very much. Good afternoon, everybody. Within the next 10 minutes or so, I'm trying to present a few practical aspects of nanoparticles from a really practitioner's point of view. If I talk about nanomilling, I'm talking about processes where we start with a classical NCE, which is a crystalline material typically, which we convert into a nano-sized product by a mechanical milling process. So this is probably much different to many presentations you would see here, where it's more a bottom-up approach, like liposomes and things like this. And here we have the top-down approach to really comminute the particles to improve the performance of such materials. I'm going to start by simply presenting some pictures of typical, here lab-scale equipment we use in the early stage of formulation development. That's two or three typical stages or sizes of mills we use. If we start in the very early stages of a new project, we start with a screening mill, which is shown on the very left. That's simply a vibrational mill where the product is filled into small vessels of 1 to 2 milliliters, containing 10 to 20 milligrams of drug each sample. And by vibration of the mill, the beads contained in the vessels comminute the particles down to the size we want to have finally, which is typically in the range of several hundreds nanometers. These are more or less only used for stabilizer screening in the very early stages, where we want to learn something about our product, which are suitable stabilizers for such a suspension. The next step would then be a mini mill, where the typical volume is, say, 10 to 30 milliliters, with a drug usage of 0.5 to 5 grams typically. And this is then used for the very first steps in formulation finding and perhaps for initial preclinical supplies for formulation testing in animals and things like that. And then the last stage is a pilot mill. This can be run batch-wise or in a continuous operation mode. If we run it in recirculation, we can say process volumes up to 5 liters here. And the drug needed then starts in the smallest scale, roughly at 10 grams. And then, of course, it's open-end there, depending on the volume we apply. And this is then still used for preclinical studies or even initial clinical supplies for a phase 1 study or something, if it's transferred to an GMP environment then. What does nanomilling mean really in terms of particle size? This is a particle size distribution measured by laser diffraction for a typical NCE, a crystalline product, where you can see we have really large particles, that's microns here, so we're in a range even clearly above 100 microns or so, which is quite a typical range. If this drug is now a poorly soluble compound, we're interested in improving the properties by reducing particle size, and the first thing that's typically done is a dry milling, like jet milling, but here the efficiency of the process is limited, so that's why we end in the several microns range typically. If we then shift to a wet milling process, we're able to comminute the particles even further. That means in the end, in this example here, we have a mean particle size or D50 value of 140 nanometers, and that's a very typical size for such a process then. These are some SEM images of typical, say, micro suspensions on the top. Oh, sorry, that was the wrong button. Micro suspensions here on the very top for 3 different APIs, and then the respective nano suspensions on the bottom. You can see, or not very clearly from this bar here is 200. It's 200 nanometers. So these are needle-shaped particles. These are more round, or these are even nearly spherical particles. So we have all different kinds of shapes of particle, but still we have a clear particle size effect by this nanomilling, starting from here and going down to here. And this is the first objective we have in these processes. It's a wet milling, so we're talking about suspensions here. In many applications we are not able to apply suspensions though, or we don't want to apply them, or if we're aiming at an oral product for human use, for reasons of stability or instability of such suspensions, we need to transform or to convert such formulations then into a solid product, and that can be, for instance, done by a spray drying process. Again, some SEM images showing examples of this. Here you see the typical structure of spray-dried particles, which are fairly large, so in the range of some tens of micrometers. But you can still see the nanoparticles embedded in the matrix polymers that are typically used for such a process. The first thing we typically look at is the in vitro performance of such particles, because this is the first thing we can look at typically. There's 5 curves in these graphs showing in vitro dissolution of the same API in different forms or different formulations. The light blue curve is only the crystalline API in its original form, so it's a relatively slow dissolution here, which we wanted to improve. If we form tablets out of these, the dissolution even becomes slower, which is quite typical, of course. If we then go to a nanosizing, so a nanosuspension, you see here it's nearly initially rising up to 100%. And staying at this level. If we spray dry the suspension, we lose a little bit of the performance, which is the red curve, but still we're able to increase the level of dissolution obtained after 2 hours here. And if then the spray dried product is even formulated as a tablet, again, some performance is lost in the first few minutes, but still the superior performance compared to the original drug or the tablet containing the crystalline large particles is obvious here. Even more important, of course, is not the in vitro performance of such products, because clearly if a drug is poorly soluble or has problems in dissolution kinetics, then we expect to see an effect in vitro. This graph is showing now a typical example of a poorly soluble drug that's applied in an animal model at different dose levels, which are given here. So starting from 3 mg per kilogram up to the high dose level of 300 mg per kilogram. And you can clearly see that especially in the high dose range, there's not a lot of difference in the PK profile here between the 100 and 300 mg per kilogram and the 300. And this may be a problem, especially in the tox characterization of new drugs. So we have a leveling here. We are not able to achieve the exposure of the drugs we want to have. And here again, nano-sized products may help and come into play. This now is more or less the same graph now comparing different formulation approaches that are typically tried if we experience such problems in the preclinical studies. The first one is the black curve on the very bottom here is now at this high dose level of 100 mg per kg is the crystalline suspension of the material. Then we have a silica gel embedded API, which is a little bit better, but still too low for this purpose. Then we have a nano suspension giving a much better performance. Only superior is still the HB beta cyclodextrin formulation, which is a solution of the API. So this clearly is a difference, but we often experience problems with solubilizing excipients like cyclodextrins in the high dose range in the applications for for toxicological characterization of the drugs, because of course, for example, 10% HB beta solution brings a lot of excipient in the animal besides the drug, which is then the cause for potential unwanted side effects, and it doesn't allow to characterize the pure drug. For this reason, especially our colleagues in tox are very much interested in those nano suspensions very often. And here's another example. Again, a poorly soluble drug. You see over the whole pH range a very low solubility, which gave exposure problems with the standard vehicles. The first profiling was done with HB beta again, but if it came to the tox profiling, a nano suspension, which is shown down here, and self-emulsifying drug delivery system were compared. Both in the end showed roughly the same in vivo performance if you look at the Cmax and the AUC values. But a clear difference is that the amounts of excipient you use for the self-emulsifying system are here 20% in the vehicle, and here in the suspension we have only a little bit more than 1%, which is a clear advantage. Nevertheless, in this case, the SATS formulation was still used for the tox formulation. The reason was the ease of manufacturing of this kind of formulation. But we know there's a lot of species-specific limitations for such systems due to the large amount of excipients that are needed. So with this, I come to a very brief summary and some conclusions. I try to show that nano suspension can be obtained by a wet milling process, ending up with particle sizes typically in the range of several hundred nanometers. These processes can be scaled from the very small scale, screening scale, to lab and pilot scale, but also to full-scale commercial. I didn't show this here, but there's a lot of manufacturers for devices or for equipment allowing to produce really in a large scale. The main in vitro effect, of course, or the main effect expected for such formulations is the improvement of the dissolution kinetics, but I try to show you some examples where we see a clear advantage of those systems also in vivo. And one of the most specific advantages of nanosuspension from my point of view is, especially in the high dose range, the very low amount of excipients that's needed to formulate those APIs here. Thank you very much.
Thank you very much. Do we have one question? Patrick.
Thank you very much for this presentation. I have a question. The milling, I assume that there you have metal surfaces acting on these materials.
Not really. Okay, I didn't show the real process. It's a bead milling. That means, of course, the milling chamber contains some metal, but usually it's a wall made of ceramics, and also the milling beads, which is small balls of a typical diameter of several hundred microns, are made of ceramic material.
Yeah.
In case of the nano suspension, of course it's then only applicable for drugs in which, let's say, in the whole absorption process, dissolution is the rate limiting factor.
Right.
That's true? Or does it have additional advantages?
No, in principle you're right. That's the main effect. But very often we have a combination. So if we find poor solubility, or we get the information the drug has a poor solubility, we very often see the combination that also dissolution kinetics is very slow. In that case, actually, it's not always possible to tell if the solubility data we get from the colleagues in the characterization groups are real equilibrium data, or if it's simply so slow that they're not able to measure equilibrium solubility. In such cases, it's imaginable that, of course, by improving the kinetics, you can improve the levels of then adsorbents in vivo also.
Very last question.
There was a huge difference in the Tmax in your example. 8 hours versus 24 hours for the sets. Do you have an explanation for this?
Yeah, perhaps one explanation may be still the dissolution kinetics, because in a SATS formulation the drug is more or less it's dissolved in the system. That's a clear advantage, of course. But then, of course, you have to see if the Tmax is relevant for that characterization, and very often in the tox profiling, AUC values are more important. But of course, there may be other differences, and it's not the whole story to compare only say Cmax and AUC. You're absolutely right, but still sometimes if nothing else works, it's the last anchor the colleagues get to use such a suspension, especially if we need to go to very high dose levels where the excipients in the SATS formulation really may become a problem.
Unfortunately, it's always the same. What I experience also, if it fails in the tox, nobody would give it to the clinic. But yeah. The question is how can this be easily transferred from tox to a clinical study? I mean, not to go with nano.
Yeah. We always, with the polysolid drugs, we typically always use or look at the nano sizing, but then the aim may be a bit different. In some cases, we are also interested in the nano sizing for the human formulation approaches. Then, in other cases, we are only interested in this for the reason of tox profiling, where we clearly know a nano suspension, for what reason ever, is perhaps not possible for a human application. So there's different cases, but in the early stages of the formulation development, we try to cover both aspects there. And yeah, there we clearly have more in vivo data. That's why I presented such examples there.